It's my great pleasure to welcome and introduce today's speaker, Professor Daphna Joel. So in this today's Distinguished Lecturer Series talk. Daphna Joel is from the Tel Aviv University, where she also studied and did her PhD in psychology. And during the first 10-15 years of her career as a neuroscientist and psychologist, she actually did some quite basic animal neuroscience work, studying the organization of basal ganglia thalamocortical circuits, underlying behavior, and trying to establish and studying animal models of obsessive-compulsive disorder. And then after 10-15 years working on this, she kind of switched fields or moved direction of research, and is now focusing more on how the brain relates to sex and gender. And in 2015, she published a very influential paper in the journal PNAS on the brain mosaic. So in 2015, Daphna published a very influential paper on the brain mosaic, basically claiming and showing that there is not really such a thing like a brain, male brain or female brain, but that really everybody's brains are really more like mosaic, or best understood as mosaics, containing both female-like and or male-like features. And with this work, she also earned this quite prestigious ERC Advanced Grant in 2021, where she continues the studies and also moves beyond neuroscience and takes this as a more general psychology question on how to study actually and how to understand gender and gender as a mosaic. So I'm very much looking forward to this talk. I think this is again a talk bridging very nicely different research fields in our CRC. So thanks for coming over from Israel, and looking forward to it.
Thank you. So thank you very much, and it's a pleasure and honor to be here. So yes, I will talk about rethinking sex, brain, and gender from binary to mosaic, and focusing mainly on the brain, but also talking about sex and gender. And before we go beyond the binary to the mosaic, let's start with the binary. So, okay. So within the binary framework, the taken for granted starting point is that there are male and female brains. And this dictates the research method, which is to look for sex differences. And when these are found, the interpretation is that indeed there are male and female brains. So we have this cycle that we start from assuming that this is true and then concluding that this is indeed true. And just to show you how it works, let's look together on the comments of an anonymous reviewer who wrote, "The usefulness of the dichotomization into male and female brains stems not only from its biological reality." Let's stop here. So for this reviewer, the question of the male and female brain is not a question at all. It is biological reality. So this is the starting point, that there are male and female brains. So the reviewer continues. So the usefulness stems not only because it's real, but also from the fact that there are many sex differences in the human brain. This is what it says here. And the collective evidence, that is all the differences that we already know that exist, still overwhelmingly warrants categorization of brains as belonging to different, if often overlapping populations, that is, as female and male brains. So the reviewer starts from the assertion that there are male and female brains and concludes that indeed there are male and female brains without ever considering any other possible relations between sex and the brain. And we can see this self-maintaining belief system also in the very famous figure, which I think those of you who are interested in the brain are familiar with. So this is a study from about 10 years ago, when they studied the human connectome, and they assessed the strength of over 9,000 connections in the human brain. And they found differences in several dozen connections. Now, you will not be surprised to learn that the title of the paper was not the connectivity in male and female brains are extremely similar. They focused on the differences, but we will go with the differences. So the differences are about in 1% of the connections. This is the focus, but we will go with this focus. And this summary diagram presents all the connections that were stronger on average in males compared to females in the upper panel, and all the connections that were stronger on average in females compared to males in the lower panel. The color code is blue for interhemispheric connections and orange for intrahemispheric, blue for intrahemispheric and orange for interhemispheric connections. And when you look at these images, and also this is how the researchers treat these images, you think that the brains of male and females are extremely different, right? Blue and orange. Men are for Mars and women are for Venus. Now we understand why. And also what you think, and this is what they assume, is that what is typical of the brains of males is to have male typical connections, the upper panel. And what's typical of female brains is to have female typical connections. This is the lower panel. Now, this logic is very familiar to us, and it is familiar from the reproductive system, where it is indeed correct. So what is typical of the reproductive system in males is to have only male typical features or organs. And what's typical of the reproductive systems of females is to have only female typical organs. There are a few people that do not have only female typical or only male typical genital organs, but they are not termed male or female. They are intersex, and it's quite a rare condition. Okay, so they take the logic of the genitalia or of the reproductive system, and they impose it on the brain without ever thinking whether this is indeed correct or without ever checking this. So they just borrow the logic. And the reason to suspect that this logic does not apply to the brain is that there are many studies in animals showing that what is typical of males and females in the brain may be different and even opposite under different conditions. And because this is so different from anything that we know about sex, I want to look at this closely, or see this closely looking at a single study. So this study is not my study, it's from Shores et al. Many years now ago. And they looked at sex differences in the rat brain. And I will now be talking about rats, and when we go back to humans, you will notice, but I will also say this. Okay, so they were looking about the density of dendritic spines in the rat hippocampus, and you can easily see a sex difference here. So the density of the spines is much higher. The spines are the small extensions here from the dendrite. So the density in females is much higher than in males. A sex difference. Now they had another group of rats, and these rats underwent 30 minutes of stress one day before their brains were assessed. And again, you see a sex difference, but it is reversed. What we thought was typical, or what was typical of females, high density of spines, we now see in males. What was typical in males, we now see in females. And as you're very quiet, and I don't know whether you really got it, so imagine what you will feel or think if I told you that instead of talking about the brains of rats, I told you that 30 minutes of stress are enough to change your penis into a clitoris or your clitoris into a penis. Okay, now you're getting it. So this is what they found, but only for the brain, okay? And I started to look for other studies like this because I thought, okay, this is very different. And I found many studies showing this reversal of sex differences in many regions of the brain, for many different features, things that you can measure in the brain, and following many different manipulations. Even housing rats, one to a cage or several to a cage, is enough to reverse some sex differences in their cortex. And when you look at all of these studies together, as I have done, what you realize is that these interactions of sex with other factors, such as stress or housing conditions, can be different for different brain measures. So that the same manipulation can reverse, for example, a sex difference in one feature. So you can see here that there was a sex difference in no stress, but following 3 weeks of stress, the sex difference reversed. So this is like we saw before, but for a different measure. This is CB1 receptors in the hippocampus. So the same manipulation that reversed this sex difference in one feature actually abolished the sex difference in the density of CB1 receptors, but in a different region of the hippocampus. Okay? So this is a general idea that the same manipulation does different things in different regions. And we were recently able to demonstrate these 2 principles. First, that on the left, that sex differences may be opposite or different under different conditions. And the second, that these interactions may be different for different brain features. We were able to show this in a single study. So what we did is, together with Alexa Winema and her ex-students Caroline Smith, they had 2 studies from Winema's lab using the exact same strain of rats, same age, same housing conditions, same food, everything is the same. And in the 2 studies, they measured the same brain measures. So they measured the density of oxytocin and vasopressin receptors in many brain regions. And there was only one difference between the studies. In one of them, the rats underwent some short social manipulation. Of course, there could be other differences that we are not aware of, okay, but at least one that we know of. So, but basically, it's very, very similar as you can get. And what I'm going to show you is the size of the sex difference in each of these regions in the 2 studies. And because it will be many, many numbers, so I'm going to use this color code. So if there is no difference, then it will be white. And if it was larger in males or higher in males, blue, higher in females, red, okay? Ready? Okay, this is what they got. And so first, let's look at the conclusion on the left. Okay, that sex differences may be different under different conditions. And you can see that in most regions, the sex difference is not consistent. Okay, so it's actually rare to see that you have the same sex difference in the 2 studies. It's much more common to have different sex differences. They may flip, they may be non-existent and then appear, or the other way around. Okay, so this is the first idea, that sex differences are rarely consistent in cross conditions. Okay, and the other one is that the interactions are different for different brain measures. So you can see that it's not that you have the same change across all regions. In some regions, it stays the same. In others, it flips. In others, it disappears. So in every region, something else is happening. Okay, so you can see the 2 general principles that up until now I was able to deduce only from studies testing 1 or 2 brain features. And here we had many, and we can show the 2 principles and also see that they are very common. So it's not that it's rare that sex differences reverse. It's actually the other way around. It's very rare that they remain consistent, okay? Okay, so what can we learn from all of these studies? First, that there are sex differences in the brain, at least in the rat brain, and we'll see later that also in the human brain. This means that sex-related variables affect the brain, affect many features of the brain. Receptor density, structure of neurons, connectivity, many, many different aspects of the brain. Now, these studies do not reveal the mechanism of these, so which sex-related variables are relevant, but other types of studies, which I will not talk about here, but which we have reviewed also, but others did too. So we know that sex-related hormones, like testosterone, estradiol, progesterone, they all affect many measures of the brain. And of course, there are other sex-related variables. Some relate to the maternal behavior, so how the mother treats the pups, and this can affect sex differences, and many, many other features can cause sex differences in the brain. But what we all also know or learn from these studies is that these sex-related variables interact with other variables when they affect the brain. And the result of these interactions is that what's typical of males and females may be different, even opposite, under different conditions. And this challenges the sex binary, and specifically the assumption that what is typical of the brains of males is only male typical features, and what's typical of the brains of females is only female typical features. Because even if I assume that at some specific point, let's say now, my entire brain is in the female typical form, so everything is pink, then following some kind of event, stress, whatever, some of my brain features will change their form, others will not, and I will end up with a mosaic brain. Mosaic in terms of having both male typical and female typical features. Okay, is this general idea clear? Okay, so this is a mosaic hypothesis I formed many years ago now, already in 2011, on the basis of animal studies of the type that I showed you here. And it took me a few years to get to the study that you mentioned in the beginning, the one with the human brain. So from now on, we're going to talk about humans. And what we did here is, together with Professor Yaniv Asaf, who is an expert from Tel Aviv University in imaging of the human brain, we looked at the structure of over 1,400 human brains. And we used MRI images, and we derived everything that you can derive from an MRI image. And we analyzed it. Again, you can read the full analysis in the paper. I'm only going to talk about one analysis in which we use VBM, just for those who are familiar. The other one's really not important, to assess the volume of 116 regions of gray volume in each brain. And we wrote this, the volume of each region in this table. So we have an Excel file with each line representing a single brain, 116 regions. And then we did many things, but I'm only going to show you one. To appreciate similarities and differences between the brains of men and women, we used the color code. So we looked for each region, and if this region was relatively large compared to the volume of this region in all other brains, men and women combined, then we painted it green. If it was relatively small, we painted it yellow. Okay, so we do this for the entire set, and this is what we got. This was an adequate response. So the brains of men on the left, brains of women on the right. Each line is a single brain. Okay, there are 116 features. And first, you don't need statistics to see that there are differences between the two sides. So at the group level, there is more yellow at the men's side and more green at the women's side. This is a known difference when you use VBM, as we've done. Women on average have more gray matter. This is why they have more green. Green is larger, okay? But at the same time, you can also see that brains are rarely all green or all yellow. Instead, each brain is a unique mosaic, unique combination of green, which is more common in women, and yellow, which is more common in men. So I want to stress that the question for me is not whether there are sex-gender differences in the human brain. There are, we showed it, others found it. They are much smaller than what you see in rats, but still we can find differences. Also, I'm not at all interested in the nature versus nurture question. I mean, we can discuss it later, but for me, I don't care why we see these differences. I only ask whether these differences add up consistently within each brain to create male brains and female brains, as is almost always the case for the genitalia, and as has been assumed to be the case for the brain, or whether brains are mosaic, as the rat data suggest. And I think that when you look at these images, the answer is clear. Brains are not yellow or green. Also, they are not orange or blue, so we also analyzed connectivity data similar to what they have done, and we didn't look at several dozens connections, but only on 7 connections. 7 that showed the largest differences in the human brain in the sample that we had. And there wasn't even a single brain that had all connections in the male typical form or all connections in the female, in the other typical form. Instead, most brain had both male typical and female typical connections. So I can safely say that not only that this does not represent the typical male brain and this doesn't represent the typical female brain, but actually that there is no person with such a brain. These brains, they do not exist. Instead, what each of us has is a unique combination of blue and orange connections. Now, when I look at these images, or when I looked at these images, it's not just that I can appreciate that brains do not come in two types, green and yellow, but also it actually seems that we all belong into a single, highly heterogeneous population of human brains, in which what is typical of humans is to have this combination of green and yellow features. And something else that you can look or see here is that there are some rare brain types. For example, yellow brains are very rare, so there are very, very few people that really have a yellow brain. But of the people that do have a yellow brain, there are more men than women. So in general, the hypothesis or the description that I wanted to test is whether this is a correct description, that we all come from a single highly heterogeneous population in which what is typical is to have this mosaic, but some rare brain types may be different, or the frequency or prevalence of some rare brain types may be different in men and women. And together with two professors from my university, Isako Melechson, who is a professor of statistics, and Amir Averbuch, who is a professor of computer science, we used several computational approaches to ask how best to describe the relations between sex categories and the brain. So again, I just want to remind us what we know. So we know that there are sex differences. We know that sex-related variables affect the brain. But we also know that the binary framework is completely inappropriate. So how should we think of the relation? So it's not binary, but what is it? Okay, so this is our task now. And again, several students that worked with us. And the first analysis was using anomaly detection to answer the question whether the brain architectures that are typical of women are the same or different from those typical of men. And I'm using this image because I'm a very visual person in the way I think. So this holds for us the assumption that the brains are either are binary, right? Male brains and female brains. And this is a summary diagram. This is real data, okay? And this holds for us this idea that we are all part of a single population. So let me explain anomaly detection. In anomaly detection, the principle is that you give an algorithm a set of data, and the algorithm learns this data. And then you give the algorithm new cases that weren't in the training set. And for each case, the algorithm needs to decide whether it belongs, whether it's normal. So it's similar to the training set, or whether it's abnormal. And there are no criteria what's abnormal. And it was originally developed for detection of viruses and worms in a computer. So we are all familiar with the antivirus that knows specific viruses and is looking for them. But if you want to protect your computer from unknown viruses, something that has not been even developed yet, then you will use anomaly detection. So it will learn your normal computer activity, and then it will be running at the background. And if it detects an abnormal activity, it will alert you. And maybe it's a virus, but maybe it's some new app that works differently, okay? So this was the idea. Okay, so why is this relevant here? So what we did is we train the algorithm on the half of the brains of one sex category. So let's say we had 1,000 women. So we took 500 women, the brains of 500 women, and gave it to the algorithm to learn what's a normal female brain. And then we gave the algorithm new brains of women, so from the other 500, and brains of males or men, obviously were not included in the training set. And the algorithm had to classify each brain, normal or abnormal. Okay, now of course then we do everything the same, but just train on brains of males and test on brains of males and females. What would be the predictions? If the typical male brain is different from the typical female brain, then if I train on brains of males, then the algorithm will classify many more brains of males as typical or normal compared to brains of women. That's clear? And of course, the other way around if I train on brains of women. However, if we all belong into a single highly heterogeneous population of human brains, then it doesn't really matter whether I train on brains of men or women. In both cases, I will be training on human brains. So the proportion of men and women classified as normal should be similar. So we have very different predictions. We won't need statistics. Very different predictions of the two approaches. Just to make sure that this system or this approach, analytic approach, works with biological data, we first tested or used as a positive control the facial morphology of two primate species. So obviously two populations, two primate species, two populations. Let's see if the algorithm can detect this. And in whatever I will show you, we analyze the data both in the high dimension space. Here it's 190 features. In the brain it will be 116, the same that we started with. Okay, so very high dimension space, and following different methods of dimension reduction, both linear and nonlinear. The whole point is that just to make sure that whatever conclusion we will reach, it's not specific to some kind of mathematical manipulation of the data. Okay, so this is why you'll see it's not important. Okay, what do we find with the monkeys? So let's go over this together. So let's start with this case that the algorithm was trained on macaques, and you can see that all the data points, the runs of the algorithm, they all fall on the y-axis, meaning that the algorithm never mistakenly classifies a capuchin as normal when it is trained on macaques. And similarly, when the algorithm is trained on capuchin, all the data points fall on the x-axis, meaning that the algorithm never mistakenly classifies the macaque as a normal capuchin. So the algorithm does a very good job in distinguishing that there are two populations in the data. Okay, clear? Okay, let's see what happens with human brains. I'm sure you are very tense. Okay, so exactly the same. And this is what we see. Okay, so it's completely the opposite. Instead of falling on the y and x axis, all the data points fall along the y equals x axis, meaning that it doesn't matter whether you train on the brains of men or women, the proportion of men and women classified as normal is very, very similar. And we got the same picture with another set of data. So what this shows us is that the brain architectures that are typical of men are also typical of women and vice versa. What it doesn't answer is what about the atypical brains? So the brains that are classified as abnormal, are they classified for the same reason for men and women or not? This approach doesn't answer. So for this we used another method which I'm sure you are more familiar with, which is unsupervised cluster analysis. So again, the principle is simple. Now we give all the brains to the algorithm, but we don't give the algorithm information about the sex of the human from which the brain was, or the images were derived. And we ask the algorithm to define 2, 3, 4, up to 10 clusters which best describe variability in the data. So the algorithm does it in whatever way it does. So let's say that these are the two clusters that the algorithm describes, that best describe the data. And then we know the sex of the humans, right? So then we can assess the proportion of men and women in each of the clusters. And just this is the real two-dimension view of these brains, okay? And the two axes are the two PCA, two principal component axes that show the largest sex difference on them. And this is how it looks. So already here you can see it's not a very clear separation between the brains. Okay, again, let's look at the predictions. So if the typical male brain is different from the typical female brain, I expect that we will have at least two large clusters, one which has mainly men and the other one which has mainly women, right? This is the idea that the typical male brain is different from the typical female brain. And we don't have any specific predictions about small clusters, okay, about the non-typical clusters. However, if we all belong into a single category or population, then we expect that large clusters, which represents many human brains, so which represent typical human brains, will have similar proportion of men and women. However, in small clusters, which represent rare brain architectures, there may be, they don't have to be, but there may be some large sex-gender differences. So again, for the big clusters, forget the small clusters, for the big clusters, we have opposite predictions. Right? With the male and typical brain, we predict that one will be of mainly men, the other mainly women. We predict the opposite, that it will have, the big ones will have similar numbers. Okay, so again, very different predictions. Let's start with the easy case of two clusters, only two clusters. And again, we use different methods of dimension reduction, and we use two algorithms, clustering algorithms. You'll see it doesn't matter. Okay, let's start with the two clusters, and first with our positive control. Let's see if we give all the faces to the algorithm will know how to distinguish between a group of macaques and a group of capuchins. And you can see that it does perfectly. So all the clusters either have one, so everyone, all capuchins are here and none of the macaques, all macaques are here, none of the capuchins. Perfect separation, no problem. Okay, easy task. Okay, what happens with the brain? Again, exactly the opposite. So you can see that the algorithms, they don't always agree whether this is only, we're looking at there are two clusters. So we're looking at the break of men and women in the larger cluster. The small cluster will be just the opposite, okay? So the algorithms do not agree whether it's best to describe the brain as divided into two clusters of about similar size, or there is one large cluster and then the other one is small. So they don't agree on everything, but they do agree on one thing, that the number of men and women in each of the clusters is very similar. Okay? Opposite than the case here. Yeah, do whatever you need. Okay. Okay. And now let's look at all the results together of all the clusters, all the divisions. And what I will show you here is a proportion of men or women in a cluster, the larger of the two, okay? So it will start from 0.5, okay? Either it's equal or it's not equal, and then I will show you only the proportion of the larger one as a function of the size of the cluster. So remember that this is where we have the different predictions. And you can see that with large clusters, the proportion is very, very close to half. So the number of men and women in the large clusters is very, very similar. It gets to very large differences only in very, very small clusters. See here? And here, for example, in the upper one, the difference between men and women there is 6 times more from one sex category over the other. I don't know which one, doesn't really matter. Okay? So this is the way we predicted, and this is what you can see here. So we again get to the same conclusion that we did from the anomaly detection, that the brain architectures that are typical of humans are also or are similarly common in women and men, okay, the large clusters. And this is similar, not exactly the same, but it's similar to the conclusion from before, that the brain architecture typical of women are also typical of men and vice versa. And we also see that there could be some large sex-gender differences in some rare brain architectures. We did not test this, but this can be important for rare conditions such as autism, for example. Okay, so in our explanation why there may be more males than females that suffer from autism, and I say maybe because this is highly debatable, but let's say this is true, then this will be a result of the rare brain type that causes autism, because most people are not autistic. So the rare brain type that causes autism for some reason is more common in men than women. But this doesn't say anything about the typical male brain and typical female brain. And this is in contrast to a very known theory of Baron Cohen about autism, which is that autism is an extreme male brain, right? The extreme male brain hypothesis of autism. According to this, that hypothesis, the typical male brain is a little more autistic than the typical female brain, right? And autistic people are on the extreme side of males. So Baron-Cohen assumes that because there is a difference in a rare brain type, this says something about the typical male brain and female brain, whereas we show that this doesn't say anything, that the typical male brain and typical female brain are similar, and even though there could be differences in rare brain types. Okay, so this is where it could be important. Again, we didn't show this, so we didn't have any functional clinical data on people here. Another thing that we did is look for the chances that a man and a woman will be in the same cluster, and what we found is that the chances that a male and a female would be in the same cluster are the same as the chances that two males or two females will be in the same cluster. In other words, this means that I'm not more likely to have a brain that is similar to the other women in this group than to the brains of the men in this group. In still other words, it means that knowing my sex category says nothing about my brain architecture. Okay, and putting this all together, and this all you know, I want to talk a little bit about this. So we know that sex-related factors affect the brain. We know that. And we know that there are sex-gender differences also in the human brain. But still, sex category provides very little information on brain structure. And I just want to say a little bit about this, because it sounds like a contradiction, okay? Because we think in binary terms. So it's easier or easiest to appreciate this general principle from going back to this very simple study. So obviously, sex-related variables affect dendritic spine density, right? But knowing that you're going to look at the brain of a male doesn't help you to predict whether this male will have low or high spine density. And not because its sex doesn't affect this feature. It does affect this feature, but how it affects this feature can be opposite under different conditions. And this is why it cannot serve to predict the endpoint. Okay, so this is very important. Yes, sex-related factors affect our brain even now as we all sit here, I stand, okay? But how they affect our brain depends on many, many other variables. And this is why also our history. And this is why knowing our sex category doesn't allow us to predict each other's brain structure. And also, and this is what we've showed, also not to predict in what ways our brains would be similar or different one from the other. And I always show these images because I think it's really, really difficult to get away from the binary framework, which is very strong. And this image is very helpful because it helps us see, really see, how it can be that there are group-level differences between the brains of men and women, but still brains do not come in two types. And the reason that it's difficult to get away from the binary thinking, because it's so prevalent, is that usually what we ask, or within the binary framework, the only question is, are men and women the same or different? When different means belonging to two types. And this is why when people find even a single small difference, then they are not the same. They are different, different meaning two types. Okay? And what we see, and this is very helpful, I think, what we see is that the answer to this question is neither. We are not the same, and we are not different in meaning belonging to two types. Instead, each brain is very, very different from any other brain. And what we were able to show is that sex category provides very little information about this variability and about how two brains would be similar or different from each other. Okay, and this is very different, again, from the genitalia. In the genitalia, it's also true. We are not all the same. But knowing the sex category of someone gives us a lot of information about which genital organs we may find there and which for sure we will not find there, etc. This is not true for the brain. Okay? Okay. So this is about the brain. And let me talk a little bit about sex and about gender. So let's talk about sex. And again, I'm not talking about sex in the reproductive system, in which, although there are problems in definition, I think this is again only social. So let me just say something about this. Biologically, still, it's going to change, I think, soon. Many scientists are working on this, but still we can only reproduce as males or females. And many of us cannot reproduce. Men can reproduce when we are too young. Women cannot reproduce when they are too old, so I can no longer reproduce. And some people cannot reproduce throughout their life, and some people can, but they just don't want to. Okay, so the question of who is male or who is female is really important if you want to reproduce. Because again, up until now, we can only reproduce as male or females. And people are working on external womb and many, many things, okay? So maybe it will change, but still this is the case. But the fact that we are so obsessed with defining who is male or who is female is just because our sex category is so important socially. If it weren't important socially, then really wouldn't matter, okay? Because most of us cannot reproduce. So most of the people that you meet cannot reproduce. Either they are too young, they are too old, etc. Okay, so I'm not talking about this, about the reproductive system. I'm talking about, for example, if you study the brain, or you study behavior, or you're studying some psychological ability, etc. And you find a sex difference. So, and I'm writing here sex difference because this is also true for animals, in which I don't want to talk about gender. Okay, so you take a group of males and females, regardless of the species, and you find a sex difference in an endpoint. Usually, when this happens, we assume, without testing or checking, we assume that this relates to what Sarah Richardson from Harvard calls sex itself. So, sex-related genes and hormones. And before I say something about this, let's just look a little closer into sex itself and talk about hormonal sex. And let's just take three very famous hormones: estradiol, progesterone, testosterone. Usually within the binary framework, we think of them, and many people also talk about them, as male hormones and female hormones. And of course, this is completely wrong. So this is, again, taking the binary system and applying it to hormonal sex. Not relevant at all. First, we all have all of these hormones. And the other level of estradiol and progesterone is very, very similar in males and females. The only exception is pregnant females. They have a class of their own, not relevant, not similar to other non-pregnant females and to males. The average levels of testosterone are higher in males than in females in some stages of life. But the distribution overlap. So it's not a penis and a clitoris. It's much more overlap than that. And hormone levels are very, very dynamic. So it's not the reproductive organs, which are stable throughout life. And this is just an example. This is from mice. So each line here or each block is a different mouse. And what you see, the level of testosterone and another hormone, but it's not really important for us. So this is the level of testosterone throughout a day. And you can see the enormous fluctuation of testosterone. And they are very different between different mice, male mice, etc. So the levels of hormones are not stable. They are changing all the time. We are used to think about the changes in females of estradiol and progesterone along the estrous cycle in rodents and the menstrual cycle in humans. But the changes in testosterone are much larger than the changes in estradiol and progesterone. And these changes are not only internally driven, but they are also responsive to the environment. So testosterone, for example, is sensitive to competition, to sexual thoughts, to parenting. And all of these features are gendered. Who can compete? Who should have or is allowed to have sexual thoughts? Who takes a major share of parenting, etc. Okay? So if you take all of this together, then obviously this view of male hormones and female hormones is completely wrong. Each of us here has a very different profile. It changes throughout the day, it changes throughout the month, and we cannot think of males and females as belonging to two classes in terms of the hormonal profile. Okay, I'm going back to this diagram, and we started with a sex difference in whatever we are measuring, and we said usually we immediately think, okay, this has to do with hormones or with X and Y chromosomes, etc. But it can be any sex-related variable. And there are many, many, many sex-related variables, especially in humans, but not just in humans. So it can be many physiological variables that are different in males and females. Some are different because of sex, but others are different for other reasons. And of course, it can be environmental differences. Again, some are driven by sex. For example, different parental treatment of male and female pups because of the testosterone, the smell in the urine. Okay, so the mother treats the males and females differently. But other are experimental derived, like housing conditions, etc., etc. So there can be many environmental factors. And of course, in humans, there is gender. Okay? This huge concept of how we are treated, how we are expected to treat, etc., etc. So all of this can be driving the sex difference in an endpoint. You don't need to take a photo. We, together with Peg McCarthy, we published this. And even more confusing, all of these other factors can affect sex itself. So I already talked about competition and parenting. They can affect sex itself. So we have a very, very complex system of many, many variables that by no chance add up consistently into males and females. So you take a group of people, divide them into two according to their reproductive system. This you can easily do. Okay, most people will fall under one of these categories. But then we usually assume that sex and gender also align this way, but they don't. There are mosaic in whatever when you go beyond the reproductive system, you start to get this mosaic. Okay, so this is about a comment about sex. Now about gender. So I already said gender has many dimensions. We can talk about gender identity. We can talk about the society, how it treats us differently. So it doesn't really matter what I can define myself as a man, a woman, a queer. I don't want to define whatever. But when people see me, they see a female and they treat me this way. Okay? So how society treats me, or how society is organized. This is one level. And of course, there is the intrapersonal level in addition to gender identity. And this is what usually been, in the beginning of thinking of gender, this is what people meant in gender. So femininity, masculinity, etc. So I'm going to show you a study studying gender at the intrapersonal level. So the relevant data is differences between males and females in cognition, personality, behavior, interests, etc. I guess you know that the most differences in cognition, personality, emotional capability, etc., these differences are either non-existent or very, very small. There are larger differences in humans in some behaviors and in some interests. Okay, so for example, most women would think that males are more sexually attractive than females, and the other way for males. Okay, so we have some large differences in preferences and behaviors between males and females. Again, I'm not interested in why we see these differences, but only in whether they add up to create two types of humans, women and men. Or maybe they are mosaic. Okay, so this is, again, I'm going back to this question. And we analyzed in the same famous study, we analyzed the behavior, preferences, etc. of over 5,500 humans from 3 data sets. And in each data set, and I'm going to show you only one, they measured many, many things. So we looked only at the variables that show the largest differences between men and women in that sample. So for example, in the study that I will show you, there were differences in these variables. And for each of these variables, there were 7. Not all are listed here. So for each of these variables, we drew the distribution of males and females and defined a feminine zone and a masculine zone. And in the middle, it's a neutral zone. So it's neither feminine nor masculine. So just an example, if someone was really worried about their weight, then there will be red, and if they didn't, they would be blue, because in that sample, this was a gender difference we found. And if someone was really, had really high self-esteem, then it will be really high self-esteem, blue. And if it was low self-esteem, it would be pink. And again, we color this according to the gender differences in the sample. But something that you may want to know, that usually when you do find a gender difference, it matches some stereotype. The reverse is not true. So there are many stereotypes that are not backed with data. But usually when you do find a difference, it will fit some stereotype. Okay, so this is why you can guess what we found. Okay, so we did this, and again here for each, the same principle. So each line is a single person. Here not 116 features, only 7 features, colored according to femininity or masculinity. Again, you can see the group level differences, more blue at the men's side, more pink at the women's side. You can also see that there is quite a lot of pink in the men's side and quite a lot of blue in the women's side. And this is because for any known psychological variable, there is some overlap between men and women. So some men are feminine and some women are masculine. Okay? This is known. We were not interested in this. We wanted to know whether there are pink and blue people. There aren't. Okay? So most people here have a combination of pink and blue. Some have only pink and white. Some have only pink and white. But there is no one that is 7 characteristics only pink or 7 only blue. And I guess you know this about yourself. So I guess everyone in this room can name a few masculine and a few feminine characteristics of yourself. What maybe you didn't know is that everyone is like this. And we've recently launched this website, which I hope at least some of you tried already, in which you answer a set of questions or features that typically show a large gender difference, at least in Western countries. And then you get your own mosaic. So this is my mosaic. So you see how masculine, feminine, or gender neutral I am in each of these different variables. And you can do your own. And the way we defined what's masculine and what's feminine here is according to a sample that we had of Americans. And obviously what's feminine and what's masculine changes across cultures. So if instead of comparing myself to Americans, I compare myself to Japanese, my mosaic changes. Compare myself to Israelis, my mosaic changes. I think there is also a German sample there, so you can see. So you can play with it, okay, and appreciate your gender mosaic and how it changes. And obviously, I'm not the only person who is mosaic, so everyone who does this will get their mosaic. And then an interesting question, which I will not have time to discuss, but I want to leave it with you, is, or maybe we will discuss it later. So if everyone is mosaic, why, when I look at this room, and you can look around on the others in this room, why do I see people as men and women? Why do I see people as if they belong to 2 categories instead of seeing them as their own unique mosaic? And the answer, and we wrote a paper about this, explaining everything that I will say in a sentence, the answer is similar to what we saw before with the brains. So we have a gender binary system that thinks that whether you are male or female is really, really important to every aspect of your life. It treats us differently according to this, etc. And this binary framework now for gender leads to labeling of people, labeling of activities, etc. This leads to many cognitive biases. And this leads us to perceive people as if indeed they belong to 2 systems. So we see the same as we did for the brain. Remember, in the brain we started with the belief that there are male and female brains, and then we never considered any other possibility, looked at the data and concluded, yes, there are male and female brains. So the same is here. We start from the assumption that there are men and women, that they are essentially different, they belong to 2 types, and this leads to many cognitive biases and many effects on the environment, which maintain this circle. And one component of this circle is a biological essentialist binary view of gender. And of course, the myth of the male and female brain, it plays an important part in this biological. And this is why people are so obsessed with the myth of the male and female brain and really want to read about differences in the brain, etc. Because this gives them this feeling that this is true. This is, indeed, we are so different. Even our brains are of two types. Okay, so we end here, and we have lots of time for discussion. Thank you.